So good afternoon, everyone. I'm Nick Gardner, the Chief Operating Officer at the BIA, and I'd like to welcome everyone to today's BIA webinar on risk management mitigation via a dual sourcing approach. So thank you all for joining us. Before we get started, please note that everybody is muted, so please write any questions you have in the questions box within your control panel, which is at the bottom right, and we will come to those at the end of the webinar. If you have specific directions towards either of our speakers, either Hans or David, do make that clear as well in that box. So the BIA, we'd like to thank Exmoor Pharma Concepts and Purolite Life Sciences for organising this webinar, and welcome to both of our speakers today, Dr. David Vincent, Bioprocess Engineer at Exmoor Pharma Concepts, and Hans Johansson, Global Applications Director at Purolite Life Sciences. Thank you for your work in putting the contents of this webinar together, and we're very happy to have you both here presenting today's webinar. So, as I am not the expert in this scenario, I'm going to hand over to David Vincent and let him present his slides. So, David, over to you, sir.
Thank you very much. So just as a brief introduction, so my name is David Vincent. I am a bioprocess engineer at Exmoor Pharma Concepts. I have around 10 years experience in biopharma and cell and gene therapy process development, particularly around the viral vector process development. So I've worked with a large number of different small and large viruses from a range of applications. Okay, so I just wanted to give you a little bit of an introduction to Exmoor Pharma Concepts for those of you who are listening who haven't worked with us before. So we are a technical and strategic consultancy in the biopharma and cell and gene therapy space. And we've traditionally focused on process design, facility design from requirement through to concept, project management during the design and build phase, all the way through to licensing, and also GMP compliance and all of the regulatory and QP certification around translational therapies from research through to commercial manufacturing. However, about a year ago, we decided to invest in a couple of process development labs so that we can now establish client processes at small scale in our facility. We can do optimization development, we can do robustness testing. Currently, we can scale from 1 liter all the way to 40-liter stir tank reactors. We also have the ability to do planar culture. We can do some stability analysis, particularly around viral vectors, and also analytical development and GMP qualification. So what we feel is one of the unique aspects of the process development lab is the link between the lab itself and the rest of the consultancy business. So we're developing processes from early stage research all the way into commercial manufacturing. But we're doing so with the end goal in mind. So we have a deep understanding of how GMP manufacturing should work from a regulatory, from a facility design, and also from an operations perspective. So we're situated in Bristol. So we have taken 2 50-meter squared laboratories with the option to expand, and it's situated on the University of West of England campus, like I said, near Bristol. And on the bottom left-hand side of your slide, you can see one of the 3D diagrams that our architect put together for us. When we were in the concept stage of designing the labs. I'm pleased to say that it's very realistic. Okay, so from a regulatory perspective, one of the main goals of an organization, whether developing drugs for cell and gene therapy applications or from a biopharmaceutical perspective, is to obtain market authorization for your product and a process, and then to continue commercial manufacturing across the product lifetime. And in order to do that, it's imperative to demonstrate continued compliance with all of the regulations. Now, part of what is required in order to do that is to identify and validate the critical components within a process. But in our experience, particularly within the cell and gene therapy space, which is still maturing. A large number of organizations are becoming increasingly reliant on single vendor sourcing for one or more of those critical components. And you can see on this slide that I've listed a couple of examples. So media, certain supplements, from an upstream perspective, certain bioreactors and production systems, chromatography resins, but also analytical, consumables, raw materials are often single sourced. And this, in many cases, we've seen had to result in extended lead times, which can lead to delays in the supply chain, and of course add costs, particularly at a late stage in process development. And this adds risk. So the question becomes, well, how do you mitigate that risk? So we would argue that you'd need to put into place a process development plan early on in the lifetime of a product, so right at the process design stage. This will then identify what the critical components of a process are, each one of which can then be risk assessed. And that risk assessment will then identify which of those critical components are single sourced. And then once that analysis has been completed, we can start to work through mitigation strategies with you. And one of those mitigation strategies is the potential to dual source certain critical components. If that's not the case, then sort of severity versus likelihood on extended lead times of single source components needs to start to be completed. And this is where continued communication with regulators is imperative in order to understand what the requirements are in order to make certain process changes. And once this has been completed, you can make a decision on your process design driven by your process development plan and driven by a risk analysis. So to put this into context, I want to take you through a case study, which is looking at the supply chain challenges for recombinant AAV capture and polish steps. So for those of you who haven't worked with AAV or other viral vectors, just as a brief overview of AAV, AAV stands for adeno-associated virus. It's a small single-stranded DNA enveloped parvovirus, and it's becoming increasingly popular as a gene therapy vector for a range of reasons. However, the most important being that it's relatively non-pathogenic, and it is able to transduce, so inserted DNA into the nucleus of a large range of dividing and non-dividing cell types. However, it has a number of limitations when it comes to its production process, namely that it's generally produced at low titer, and is the case for a large number of viral vectors. There are a large amount of variability and time-consuming analytics required in order to make process decisions, which becomes very difficult. But in terms of productivity, certainly from a downstream perspective, what we've seen is that low titers and relatively large volumes places a lot of pressure on the purification steps, and as relatively small columns are required in order to maximize resin utilization, which therefore has an impact on process time. So in order to capture, so the first step of the purification step, there are two options which are generally available. One is an affinity chromatography, and the other is a cation exchange chromatography system. Now there are two commercially available affinity medium available. One is AAVX by Thermo Fisher Scientific, and the other is ABB Cephros by GE Healthcare. From a cation exchange perspective, there are many more options. There are lots of different vendors supplying cation exchange resins, and a lot of different base matrices available as well. So the decision from a cation exchange perspective is whether to use packed bed resins or continuous stationary phases like membranes, monoliths, and fibers. Now monoliths in particular are becoming increasingly popular due to their fairly impressive performance attributes. So they are able to generate high recoveries, they have high binding capacities, they can be run at high flow rates, and they have an impressive resolution. But what I've tried to do here is to show you in this source matrix how easy it might be to switch from one medium to another. So you can see that there are a number of single suppliers and a number of multiple suppliers of each of these medium. And the main point that I'd like you to take away from this slide is that, at least relatively speaking, there are some challenges, but it's relatively easy potentially to switch from one affinity to another affinity step. But it's quite difficult. In fact, it's very difficult to switch from an affinity to an ion exchange system, particularly as you move towards late stage process development and commercial supply. And then switching between ion exchange medium will likely depend on the base matrix used. So if we concentrate on the affinity resins available, so like I say, there are two resins available, one by GE Healthcare, which is an agarose-based bead. The other is by Thermo Fisher, which is a polystyrene divinyl benzene, so a porous base matrix. Now, both of the ligands are relatively similar, so they're both camelid-derived single-domain antibodies. So one would assume that the absorption chemistry is relatively similar. And while there will be comparability studies that need to be performed, such as process performance, impurity profile analysis, such as the type of host cell protein and the concentration and quantity of host cell protein coming in the elution, the DNA concentration, and also empty-full ratio, which I'll go into a little bit more detail later. It's likely that both of those ligands will be fairly comparable. However, when we're switching between ion exchange systems, the story may be slightly different. There are a large number of packed bed resins to choose from. However, as the performance attributes of monoliths and membranes is something that the industry is moving towards for viral vectors, you can see that there are limited options in terms of base matrix. So most membranes that are used are cellulose-based, not all, however, but most, and also the fibers are cellulose-based. However, KIM monoliths are currently polymethacrylate. Now there is advantages and disadvantages of both of those base matrices, but switching between them is potentially challenging. Therefore, it may be easier to show comparability if moving from let's say a polymethacrylate membrane to a polymethacrylate particle or vice versa. And that's something that needs to be considered when you're choosing what type of base matrix to use in your design of your process. Moving to the polishing step, there are two unit operations that are used throughout the industry and also in academia to separate empty from full AAV capsids. So now, to give you an overview of what that means, again, for those of you who have not worked with AAV, so AAV comes in two types in a process. So AAV is a virus that has a protein capsid, and inside that capsid you'll have a single strand of DNA with your transgene. And of course that's your product, but there will be a large number of viral vectors that don't contain that DNA, and that becomes a process-related impurity which needs to be removed. So the two unit operations to do that, like I say, are anion exchange chromatography and ultracentrifugation. Now the industry is moving away from ultracentrifugation. The reasons being that it can be historically difficult to scale up, can result, particularly with small particles, in very long process times, is also very, very expensive from a capex and facility cost perspective. However, even though chromatography does outperform ultracentrifugation in many ways, ultracentrifugation is sticking around, and the main reason for that is it requires a fairly small amount of process development. So what I've done here is again tried to put this into a source matrix. So on the left-hand side, you've got your current process, and along the top, you see the process change. And what I'm trying to show from this slide is that while there are some challenges to switching from monoliths to particles, or maybe from monoliths to membranes within an ion exchange system, at late-stage process development, it's going to be very difficult to switch between chromatography and ultracentrifugation. So that decision needs to be made from the offset. Now, the reason for that apologies. So the reason for that is that ultracentrifugation and chromatography are utilize very different mechanisms of separation. So chromatography, anion exchange chromatography is separating the product and process related impurities based on charge, while ultracentrifugation in this case is separating based on density. So when performing process comparability studies, it's fairly unlikely that the two processes will be comparable. It would be much more difficult to show the regulators that the processes are comparable. Not only that, but the facility requirements for ultracentrifugation are very different. It's very difficult to close an ultracentrifugation process. This is something that's becoming increasingly required, particularly in the cell and gene therapy space. And the requirements in terms of equipment and also cleaning validation are therefore very different, which makes it very difficult to switch between the two operations at a late stage. So in conclusion, single sourcing is a risk that needs to be identified and taken into account as early on as possible by putting a process development plan in place. And by doing this, we can generate mitigation strategies and open discussions with the regulators from the outset. And then lastly, Exmo Pharma Concepts is well placed to be able to help you with this with this step. So with process design, with risk analysis, with process development, change control, and also regulatory support. So thank you very much.
Okay, thank you very much. Super stuff. Thank you, David. So now I'm going to hand over to Hans Jansen, and I will let him go through his slides and his presentation. And remember to note down any questions you may have, put them into the requisite box on your control panel. So, Hans, over to you.
Okay, thank you. So I was kind of smiling when David said low titers and high volumes, because when I started in the biotech industry a bit more than 30 years ago, that was really the problem with monoclonal antibodies. A good titer was like 20-30 micrograms per milliliter, and you had huge volumes. Now it's very different, and I think especially in the protein A area, capacity is one of the main performance parameters that's interesting. So my talk, before I move into my scientific part, I just like to say a few words about the company I work for today, Purolite. Life sciences. So Purolite has been around for about 30 years and is a plain resin company. The only thing we manufacture is really resins. And I think they are the second largest suppliers of resins today, but until 5 years ago, the main focus was all types of water treatment and sugar refineries and minings and nuclear power plants were typically customers. But around 5 years ago, they decided to move into the life science area, and now we have an agarose-based chromatography platform designed for primarily whether recombinant protein purification and especially antibodies. We are about 1,400 people situated all over the world, while the main R&D hub for the agarose technology and protein chromatography is based outside of Cardiff in Wales, UK. And that's also where we're building a full-scale manufacturing plant.
Hello, I think we may be having some technical issues there with Hans' connection. So we're not getting any volume from you, Hans. So 2 years we'll go back to you now and just check if that's working.
We already have a range of chromatography resins. So we have 3 different protein A resins, and I'll go a little bit into detail around those. And we have cation exchangers and anion exchangers of different particle sizes. So we kind of cover the basic antibody purification platform.
If you could just bear with us for 2 minutes, we'll just contact Hans outside the forum here. Just 2 seconds, please.
I have problems advancing the slides now. It's like I've lost control over the slides. I apologize for this. I tried to get some help to advance the slides. Oh, there we are. I apologize for that. So two of the protein A resins, we have two alkaline stable protein A resins called Presto AP for production and Presto APC for clinical production. They are very similar to MAB Select Sure and MAB Select Sure LX, which is the dominating protein A resins on the commercial manufacturing as of today. And this comparison is using human polyclonal IgG, and they have very similar binding kinetics. And this has also been verified, I think, by now over a large number of different customers and probably more than 35 different MABs. It's an alkaline stable protein A and it's multi-point attached, which gives it fairly low leakage levels. So with that, I move over to the case study. Actually, it's two case studies. We did one first together with Perk and Elmer and Alvotech, a biosimilar company, who kindly supported the feedstock, a biosimilar with about 3 grams per liter. So the first study was done in a Janus workstation, so a robotic systems using small atoll columns. And in the interest of time, I won't go through it here, but if you are interested, this is published as a supplement to bioprocess engineering. And the result for this, where we compared 3 different our in-house Presto resins, was really that with this particular biosimilar feedstock, we couldn't see any difference in yield or purification performance, and very low and similar protein A leakage, with the idea that you could use the least costly resin, which is based on a recombinant protein A and a very inexpensive for clinical manufacturing, and then possibly, because only a very few, obviously only a few MABs entering clinical will actually make it to commercial manufacturing, and then you typically have a window of a couple of years where you could switch to alkaline stable protein A resins. But nevertheless, to take this a bit further, for this purpose of second sourcing, we wanted to test a larger range of protein A resins. So we used the same feedstock, but we used it in a standard column system, a small column, and we assayed for whole cell proteins, protein A, and DNA using an alpha ELISA assay. The resins we tested were the ones on the list. So the top 4 are all agarose resins, same mean particle size. The AC has a recombinant ligand. Our 2 Presto AP and APC has a C-domain based ligand. The MAB Select Sure has an alkali stabilized B domain. And then we have Toyopearl, which is a small bead, 45 micrometer, metacolate bead, also a C domain based protein A. Kaneka, which is a cellulose based bead, 65 to 85 micrometer, also with the C domain. Then we had a Procep Ultra Plus, which is CPG, controlled for glass bead with a recombinant native protein A. and finally Eshmuna A, which is polyvinyl ether beads, also with an alkali stabilized C domain. So we looked at we have problems. I seem to have some problems. Ah, shit.
Hello, apologies again. We're having sound issues. I think we might have a volume turned down on Hans' machine.
You still can't hear me?
If you could hear us, Hans, just pop that back up. It would be great. Thank you.
Oh, I hate this. Is there no sound? It comes from...
My apologies. We'll give it one more minute to resolve the sound issue, and then we'll move on.
Is there still no? I've tried.
Okay, if you give us another minute or so, we're just going to see what we can do. We're going to contact Hans separately, and we'll come back online. Please be thinking of those questions. We may address some questions in the meantime, and go from there. Okay, we've got a solution. Hans is going to dial in via old technology, and then we'll move the slides on. So just bear with us. We're sorting that out presently. Thank you for your patience, people. We are still working on this. My apologies. We will be getting it back up and running shortly. Sorry, technical issues. I'm going to blame the snow.
Can you hear me? This is weird. You can't hear me? Can you hear me now? I can't hear anyone speaking back. Wow. It's like a one-way connection then.
Okay, apologies. I think we are sorted. Hans, do you want to continue?
Oh, now I can hear you.
Okay, please do proceed. Sorry about that. Slight technical issue.
Okay, can you hear me? Okay.
Yes, all is well.
Sorry for the technical problems. So I'll continue now with the actual study. Nice. The problem is I can't advance the slide. Oh, here we are.
So we use an alpha LISA. It's a very sensitive proximity assay to look for high throughput assay. And that was performed by Perk and Elmer. We looked at whole cell proteins, protein A, and DNA, which are three of the more critical contaminants in the first step. So if we look at protein A leakage, the cause of protein A leakage is really different causes. One is the protease activity in the feedstock. So things like the temperature of the feedstock and the protease content is important. So there is quite a large variation, and I've seen protein A leakage levels from 10 all the way up to 1,000 parts per million from the same type of resin. A second source is, well, protein A is a protein, so there are deamidation, even if it's a very slow process. So we always recommend to store protein A resins under weakly acidic conditions, like pH 5, and always run a blank run after storage before you do the actual process run. And then it's also a function of the immobilization chemistry and the resin material. So we did test them for, if I come to the next slide, so we did a typical protein A run, so loading the feedstock, washing, and then we introduced an intermediate wash that contained high salt and slightly elevated pH to pH 8, and we collected that fraction, which is in the black on the left-hand side, for all the different resins. And as you can see, there are fairly low leakage for the Presto resins, a little bit higher for the single attached MAB Select Sure. When the one that stood up was Procep Ultra, but that is a bit unfair because at pH 8, PPG is not really stable, so you would never use an elevated pH for an intermediate wash for that type of resin. On the right-hand side, you have the eluate fractions. Very similar levels basically for all resins for this particular feedstock. Remember, there can be quite big variations for different feedstock, except for Procep Ultra that had a significantly higher leakage. And we looked at whole cell proteins. So whole cell proteins, of course, depend on the particular cell line. Nowadays, with the advanced technology on working with cell lines, there are some examples where I remember some scientists from Genentech actually published data where they knocked out the gene for a particular difficult whole cell protein. Otherwise, it's more common to optimize to introduce an intermediate wash to bring down the amount as much as possible before elution. So once again, on the black, on the left-hand side, you can see the whole cell proteins in the wash fraction. So that's also an indication of the type of unspecific binding you have of whole cell proteins to the actual base matrix. As you can see, all the 4 agarose resins gives fairly similar values. Then you have the methacrylate, soy pearl, standing out with having much higher values in the wash. And the other 3, Kanika, Procep, and Ashmoon, have kind of similar levels in the wash fraction. In the elevate, most of the resins gave values below the limit of quantification, and except for Toyo Pearl and Ashmoon that stood out with somewhat higher whole cell protein values in the elevate. So finally, we looked at the DNA in the product pool. DNA is really not a problem in antibody processes because downstream of protein A, in almost all processes, you will have an anion exchange that will readily bring down the amount of DNA to levels acceptable for final formulation. The only one that stood out a little bit was the cellulose-based resin, 10-cat A. So to finalize this study, we do see an impact of the matrix material on the purification performance. There are some variations. So one could consider, if you have a certain process condition, wow, this kind of moves ahead of me. It is an advantage to have the same type of base matrix if you want to dual source a protein A resin for your particular process, or even validate two protein A resins before you enter the clinical manufacturing. So we think this kind of supports the idea of switching between different agarose-based resins. You can also wonder what the impact of unspecific binding has on the functional lifetime of a resin. Because once you move into commercial manufacturing, you probably expect to be able to run your resin for at least 100 cycles. So before handing over for questions, I just want to mention that we do have some exciting projects ongoing at Purolite. We're working with a technology called jetting, where you can produce almost monodisperse or much more uniform particles than you can with the current batch emulsification technology. And we've actually already launched 2 ion exchangers, 35 micron high resolution ion exchanger for this purpose, using this technology. And if you look at the left hand of the 2 microscopy pictures, you can see the difference between the batch emulsified resin at the bottom, standard agarose bead. This could be the current Presto range, or the Mab Select or Cephalose type of resins. They would look like that. But with the jetted innovation technology, you get a much more uniform. And that has a lot of advantages, both with respect to flow and pressure performance, as well as column packing and fouling. So we are very excited about this, and we will have a small scale manufacturing facility online this month in March. And finally, I just want to show that we are also working hard the last 2 years to design and build a full-scale manufacturing plant in Llantrison outside of Cardiff, and that will be commissioned this summer and could supply up to 100,000 liters of agarose resins per year, which as of today roughly is 30% of the world's agarose demand. So with that, I would apologize for all the technical glitches, and then I'll hand over to Nick to run the questions.
Thank you very much, Hans. My apologies. Let's just blame the weather. Thank you very much to both of you. Now we'll open for questions. So do write any questions that you may have in the questions box within your control panel, and we'll get through as many as we can. If you'd like to ask something specific, please do address it to who you are directing the question to. So just speaking as a relative novice, but as somebody who used to run a clinical diagnostic laboratory, you mentioned regulatory issues, I think both of you, but how would you, or how did you address those when going through a dual process, or dual, if you have to swap from one SOP or process to another one? What were the regulatory concerns that you had to overcome?
So I think if in this perspective, you would need to show comparability. So in terms of a process or an analyticals, particularly from a process step, then you would need to have a dialogue with the regulators, and then to show them a package of so for them to be able to understand whether one unit operation and your dual sourced unit operation is comparable.
Okay.
And we've had some discussions with FDA representatives, and so in the end, obviously you have to do some type of comparability study. But if we're talking specifically about protein A, going from continuous to batch, which a lot of companies plan for to do continuous in pilot or clinical manufacturing, and then switch to traditional big packed bed chromatography for commercial manufacturing, that would probably be a bigger difference in related to the product quality than going from switching between two different types of protein A resins. But of course, regulatory authorities never give a clear answer. This is what you have to do, and if you do it, it's okay. So it's like David says, you have to have a communication discussion with the authority.
Yes, horribly familiar.
Yeah.
Okay, I think we may draw a conclusion to this afternoon's proceedings. Thank you very much to both of you, to both David and to Hans. It's been very informative and extremely interesting. This webinar will be available on our BIA YouTube channel from tomorrow morning. We'll edit out the slight technical glitches here and there, and the slide set will be accompanying that as well. So I'm also also thank you to Exmoor Pharma Concepts and Purolite as well, Life Sciences, for your time and effort in today's webinar. So it finally leaves me to just say on the screen now our upcoming BIA events, and that I think tomorrow is the end of our early bird registering for the MHRA conference, which is particularly pertinent at the present moment, but a very well attended and extremely interesting conference, which is on the 5th of July. The rest of our events are up on the screen, so if you have any questions, please do get in touch with us as normal. Other than that, thank you all for attending, and goodbye.